Okay, today we're going to be talking about cell division of mitosis and meiosis. Alright, so I just got some pictures of some real cells dividing right here. Okay, so cell division is highly organized. Cell division occurs when fertilization is occurring or when cells die and new ones are needed, or when cells need repairing, they replicate. All the stages of mitosis and meiosis occur because of highly regulated checkpoints that are controlled by molecules. So right here I have a diagram of all the phases of mitosis. So as you can see, there's phase G1, S, G2, and then there's cytokinesis and actual mitosis, and then there's three main checkpoints that I'll be talking about in this video, the G1 checkpoint, G2, and M checkpoint. Okay, so the checkpoints check for crucial events that are occurring before the cell divides, and so the checkpoints are there to make sure everything is okay before the cell continues in the process of cell division. So in eukaryotes, the cell has a system called the cell cycle control system, and this is made of a bunch of regulatory proteins that scan for key events to happen before giving the cell a go signal in order to continue with replication. So right here I have the cell cycle control system. As you can see, it just shows all the phases again, but then it also shows some of the regulatory proteins that are happening, such as like growth factors and other things of that nature. And then it also shows the checkpoints, which are labeled by those red marks right there. Right. So then the first checkpoint is called the G1 checkpoint, and it's considered the most important because it scans to make sure the cell is able to divide by looking for all the nutrients and proteins that are required for cell division. So after this checkpoint, the cell will either divide, the division will be delayed, or the cell will go into the G0 phase, which means that it won't divide. So here I have what the G1 checkpoint checks for, which is cell size, nutrients, growth factors, and any DNA damage beforehand. As you can see, it's at the bottom of the circle right here where that black line is, and it happens right before the S phase of mitosis. So then I have this little man over here that kind of gives you a visual for what the G1 checkpoint is. So he's asking, is the DNA okay, enough sources for cell replication, and things of that nature. Okay, so then there's the G2 checkpoint, which occurs after the S phase, and what it does is checks to make sure there has been no DNA damage during the S phase. It checks using proteins that scan for signs of DNA damage, and then if there is any damage, the process of mitosis is delayed so the DNA can be repaired. So then again, I have this little dude right here that gives you a visual for the G2 checkpoint. And then right here I have a more scientific way that the G2 checkpoint is done, and it shows a bunch of the proteins that are used. So it's actually, I believe, a negative feedback loop, and so the proteins, so when there is DNA damage, the proteins will say whether it needs to be activated or not, and then when something is wrong with the DNA, other proteins that can help repair the DNA are brought in, and then once everything is situated and the DNA is repaired, then mitosis can continue on. Okay, so then lastly, there's the M checkpoint, which occurs right before the cell enters anaphase, and it just makes sure that the sister chromatids are correctly attached to the spindle fibers that are about to push the chromatids apart. So here I have a diagram of anaphase to give you a better perspective about what's happening and what it's checking for. So as you can see, spindle fibers connect to the kinetochores of the chromatids, and so they're about to push it apart. So basically the M checkpoint has proteins that check to make sure that the spindle fibers are correctly attached to these. And then I have this little dude again asking the same questions. The spindle fibers are properly formed and attached, and that the chromatids are correctly oriented inside the cell. Okay, so then in checkpoints, there's internal and external factors, and these can include internal factors such as promoter proteins, and then external factors would be growth proteins that are available between the G1 and the G0 phase. But internal proteins are present in all the phases of mitosis. So then right here I have some growth factor signals, which would be considered external factors. So there would be a receptor on the cell surface in the phospholipid bilayer, and when a growth factor hits the cell surface receptor, the protein kinase cascade, the PKCs, are activated, and in the nuclear membrane, it can activate other proteins that code for cell division in chromosomes. So then that's when the cell would know how to that it's time to divide. So this is a diagram that would show what's happening between the G1 and either the G0 phase if there is no growth factors, or the S phase if there are growth factors present. Okay, so MPF stands for maturation promoter factors, and these are a combination of cyclin and CDK, and CDKs are cyclin-dependent kinases. So when they combine together, it makes an MPF, and these are used to regulate the stop and go signals of cellular division. So I have this diagram right here that again shows all the phases, but it also includes cyclin in the CDK. So there's many different types of cyclins. There's G1 cyclins and end-phase cyclins and stuff of that nature. And then there's also the CDK. And once they join together, that's when the cell knows that it can continue on with the G1, S, or G2 phase. But if those are not present, it will not continue on. And then once the MPFs are used to regulate this, then they are degraded down, and these are called degraded cyclins. And again, over here I have like kind of the life cycle of an MPF, where the CDK and the cyclin join together and then make the MPF, and then once that is used in a checkpoint, the cyclin is degraded down, and then the CDK can be reused again and join up with another cyclin. Okay, so then there's PDGFs, which are platelet-derived growth factors, and these are cell surface kinase receptors. They're important for the formation of blood vessels in cells, and so they kind of allow a cell to skip the G1 checkpoint. So here I've got a little diagram of what's going on, and PDFs have a there's four main types of PDFs. There's alpha, beta, receptors, and they're on the cell surface, and so these will come in and they're recognized by these PDGF receptors, and once the cell recognizes these, it will continue on with repairing or making new cells. Then there's cyclin and CDKs, which I kind of talked about earlier, and once a cyclin joins up with a CDK, that's when the CDK is activated, and then it can initiate processes in cell divisions by using phosphorylation. Here I've got kind of a cycle of it again, and this is similar to the one I showed 2 slides ago, where there's certain cyclins for each phase of mitosis, and then it also has its own CDK that matches up with it, and then once a cell receptor recognizes these, it would continue on with the process. Okay, so then this comes back to the topic of how cancer happens, and this is when a cell system control system is disrupted, and then this can result in cancer. So an example would be when there's an overabundance of growth factors in and around a cell, because it's an external factor. So then the cell would begin to replicate abnormally, and this causes cancer. So here I have a pretty basic diagram of kind of what happens. So normal cells, you get growth factors and the cells divide, and then you can also have no growth factors, so there's no division, and this is a controlled process. But with cancer cells, there's an overabundance of these growth factors, and then the cells divide abnormally and don't stop, and so then it causes cancer to spread. Right, and that's all.